Okay, Chuck had told me that he's got plenty to talk about, so I want to start as close to on time as possible. We appreciate you joining us for Grand Rounds today, and let me welcome you to the Department of Psychiatry Grand Rounds. Our speaker today is Dr. Charles Raison. Chuck is currently Associate Professor at Emory University for 3 more days. And then we're very, very excited that he will be joining the faculty in our department. Chuck recruitment was made possible through a nice, extensive collaboration with the Norton School of Family and Consumer Sciences, and the efforts of a group that was started here in the Department of Psychiatry with Chuck's collaboration with Dr. Karen Wise, Richard Lane, and other folks that are involved in the mind-brain-body research collaborative efforts. Chuck has done a lot of work related to the neuropsychoimmunology, the effects of stress in mental health and adaptation, and the use of different forms of intervention that range from complementary and alternative medicine to very fancy psychopharmacology and innovative immunoregulatory and immunomodulation type of interventions. So as he mentioned, he will have plenty to tell us, so I don't want to take a lot more of his time, but please help me welcome Dr. Raison twice today to the department and to Grand Rounds. All right.
Well, hello, everybody. I was giving a talk sort of like this, maybe, what, 2 years ago, 3 years ago? I think it was 3 years ago that the idea of me sneaking out here was actually born. So I'm extremely pleased to be here. So, you know, let me say a couple of things real quick before I jump into things, which first is that, you know, I spend my life studying the immune system and stress system and social processes, and basically sort of asking, you know, how do mood disorders especially develop? And, you know, any work I think should be judged by at least two criteria. One is, does it yield practical benefits? And I was going to give the practical benefits talk today, and I apologize to Francisca, who's indulged me in not doing that. We've just done this big study, as he mentioned, of giving a very unusual study looking at whether if you block inflammation, it treats depression. And the answer is very interesting and complicated and just not quite ready for prime time. I bet if I was doing this talk in 2 weeks, I could have delivered that, and I'll give that talk at the psychopharmacology at your CME event in the spring. The other thing, though, that I think that is important about ideas is whether or not they help clarify things theoretically. Do they take things that we know but that are sort of in pieces on the floor, and can they suggest novel ways of looking at things? Now, sometimes it doesn't even have to be the correct way of looking at things. I think there's a benefit in just ideas that shake us up a little bit, and that's what I want to do today. I want to talk about one of the implications of the role of the immune system and the inflammation in particular in depression, which is it actually, I think, gives us a new way of thinking about why depression exists, why it has the symptoms it does. You'll see. So that's what I'm going to talk about today. I want to give a talk about how inflammatory processes and host defense play into the evolution of genes that give rise to traits that we recognize as depression. Now, my other caveat is that this is an 8-hour lecture. I've done the 8-hour lecture twice, and I barely have enough time to do the 8-hour lecture. So you guys are going to be here actually then until midnight. It's a long. No, I've cut it down, and what I've done is I've done it in chunks, and so I'll just quit whenever I run out of time. I'll get through most of it. I'm not going to give you anywhere near the 8-hour version, but I just want to let you know that it's a challenge that way. This is a large idea. I think we've almost got it accepted at molecular psychiatry. So even though some of the things I'm going to talk about are a little bit odd, I think they're going to at least get a hearing in the mainstream fairly quickly. So let's start with this great quote by Charles Darwin, that the sight of a feather in a peacock's tail, whenever I gaze at it, makes me sick. Darwin really developed an anathema for peacocks, of which there were many in his garden, because he realized that they were a terrible challenge to natural selection. He'd seen plenty of foxes and dogs chow up these big, vain, idiotic male peacocks with these ridiculous tails. And as he worked on the idea of natural selection, he kept asking himself, how could something like the peacock's tail exist? It's such an obvious survival liability. Why hasn't natural selection culled it? Now, he did come up with an answer with that gave rise to what's called sexual selection theory, but I want to suggest to you that we have an almost identical question or problem in front of us when we think about depression, which we know to be at least partly a genetic condition. And this is the problem, which is that like the peacock's tail, which to poor Darwin seemed at the time too obvious a liability to have been allowed by natural selection, major depression, and not just major depression, mood disorders in general, strike early in life, earlier probably all the time, most often during prime reproductive years when you're going to take the worst hit to not only survival but also to your ability to reproduce. And these disorders are so detrimental to survival and reproduction, and I'm not going to show you evidence on that. I'm just going to ask you to take my word for it. The data on that are very strong and very disturbing. But the MDD is so detrimental to survival and reproduction, it's hard to understand why allelic variants, so why the forms of the genes that promote the disorder have not been culled from the human genome. Why, in fact, far from being culled, the genes that appear to promote depression are both so common and so numerous. All right? So as we'll see, when we look at the genes that have been best identified, and I say that with quotes because we really know very little, in fact, about the genetics of major depression. But when you look at what we do know, most of the genes that have been most often implicated are present in very high ratios in the human population. For example, anybody here ever heard of the short allele of the serotonin transporter? Yeah, you all know about that, right? Okay, cool. What do you think the prevalence of the risk, the S, the short allele is, say, in Japan? This is the form that causes depression. What would be a high prevalence, would you say? 30%, 40%? About 90% of Japanese have the S allele. It's so common they can't do studies there, right? So there's a mystery to be solved here. Why are the genes that cause depression, and I use the word cause again in quotations, but just for simplicity's sake, let me say that, why are they so common? Not only are they common, there's evidence that the genes that promote depression, promote bipolar disorder, promote schizophrenia have actually been undergoing positive selection over the last 100,000 years or so in human populations. Not only are they common, but they're getting more common. And I mentioned the short allele of the serotonin transporter to you here. Here it is. It turns out to be a bit of a risk gene for schizophrenia also. At least some studies suggest that. But there's very nice data that in several populations, its incidence has been increasing over recent time, right? So this is like adding insult to injury. Do you see that it raises a challenge, right? You know, why is it that something that seems to be so detrimental, why should it be increasing? Well, okay. So this is the starting point for what we call pathogen host defense theory, or pathos D, and it's from a great thinker, a guy named Paul Ewald. I'm just going to read this to you. He says, when diseases have been common in human populations for many generations, like major depression, which we know from Egyptian papyri goes back at least to 3000 BC, and still have a substantial negative impact on fitness, as is clear that major depression does, these diseases are likely to have infectious causes. These considerations suggest that the most important of the human diseases that are now thought to be caused by infection will eventually be shown to have an infectious cause. Now, these causes can be causes in an approximate sense, right? So, you know, you get influenza virus, you get the flu, or you have a tuberculosis bacterium come into your lung, you get TB. Or they can be causes in an evolutionary sense, and this is what we're going to talk about today. So, for instance, that the Plasmodium falciparum, the agent that causes malaria, is the evolutionary cause of sickle cell anemia, right? Because, of course, the sickle cell trait protects you from malaria, right? The only such compensating fitness benefit that has been documented for major human genetic diseases is resistance to infection. So, when we think about the fact that depression genes are extremely common and depression itself is highly maladaptive, it demands something called pleiotropy, which is the fact that genes can have different effects, sometimes opposite effects, right? There's a thing called antagonistic pleiotropy. If a gene exists at more than, say, 1 in 500, 1 in 1,000 of the population, it must be doing something positive, or else it would be removed from by natural selection. I have a whole part of this talk that I cut, but it's very striking. Diseases that are purely genetic and purely lousy. Those are the diseases that have 5 guys' names attached to them that you learn about in medical school, that there's 30 of them in the world, right? You know, the reason is because they can only be as common as the mutation and how long it takes the mutation to be removed. The more detrimental disease, the faster the mutation is removed. When you see a disease that has genes that are very common, those genes must be doing something positive. And of course, there's many people that have theorized about what depression might do positively for human interactions with each other, and I'm not going to talk about that today. I'm going to suggest instead, and this is the question we're going to ask, is it possible that depression and other mental disorders are like sickle cell anemia and exist because their alleles offer protection from infection? Now, I'm going to show you that, in fact, it's not exactly like sickle cell anemia. The connection between depression and infection and host defense against infection is actually much, much tighter and more elemental. But let's start with this. And this is the idea that I'm going to suggest to you today, and then I'm going to show you data for it in the rest of the talk. Here's the theory. The theory is that the risk alleles for depression originated and have been retained in our genomes not because depression is adaptive in some secret way. It doesn't make you a better boyfriend. It doesn't make you get along better with people. It doesn't help you analyze problems better. These are all ideas that people have suggested. But from this perspective, we say no. The genes are so common because these alleles promote host defense against pathogens. And moreover, that this enhanced host defense is accomplished mostly by heightened innate immune activation. That's a fancy word for inflammation, which leads to more robust sickness, which results in reduced death from infectious causes, especially in infancy, when selection pressure from infection is strongest and the adaptive immune system, your T cells and your B cells, are not yet fully operational. So this is the idea. The idea is very simple. Now, what's the data for it? Well, we have a hard time understanding, as we sit here today, how powerful a factor infection has been across not just human evolution, but evolution in general. Here is a poor ant that's had a fungal infection. The fungus has sprouted out the top of its head and has taken over its brain and is making the ant do all sorts of weird things that we'll talk about if we get to the end of the talk. But the point is that prior to the 20th century, nearly everyone who ever lived died from infection. And in most societies, in most times or places, 50% of individuals died in the first year of life. So you think about the Hopi, right, up north in the Colorado Plateau. Traditionally, they didn't name their children until they were 1 year old. They were sort of said to be on loan until that point. And because the first year of life was so dicey, it was easier to see their offspring as sort of only partially emerging into the world. Death rates are sky high early in life and very much connected to infection. There's 2 peaks. It's very interesting early in life. The first peak of death is at 6 months, and that's when antibodies that came from the mother in gestation pass finally out of the baby's body. And the second big death peak is at 2 years old, which is traditional weaning period, right? It turns out that breast milk, whatever else you want to say about it, is chock full of all these antibiotic substances that have huge immunologic effects. So because death from infection has been the leading reason for failure to survive and reproduce in humans, it's exerted an unbelievable, tremendous selective pressure on us. Anything that helped us survive, especially that nasty cut that occurs in infancy, to live and reproduce, there's a very strong selective pressure to retain anything that helps us in that way. So my suggestion is that the genes that cause depression or that contribute to depression are so common because it turns out that they help us survive infection, and that they do it actually by causing depressive-like symptoms. That's what I'm going to show you. Now, today I've only put up 4 of the various propositions that should be true if this idea is true, and I want to show you some of them. So let's start with number 1, that inflammatory activation should increase depression, and depression should be associated with increased inflammation. So if I'm saying that the genes that cause depression have been retained because they help you survive infection, and they do that mostly by increasing inflammation, then by golly, inflammation should make you depressed. And people with depression should have inflammation increases. We and others have shown, this is data from Emory, that you can take people that are perfectly not depressed and relatively healthy. This is a population of people that had a single melanoma lesion. They had no metastatic spread. If they had any depression, they weren't allowed into the study. So these were people that they've all died. They all died within 2 years of entering the study. So we took all the unreasonably optimistic folks who thought they were going to beat it and who were not depressed, and they all went on interferon alpha. In this population of people that were actually fairly resilient, 50% of them will developed a full major depression in 3 months. So 3 months of chronic inflammatory activation, 50% get a major depression. Now the interesting thing about this study was that out here, before they started the interferon, people were randomized, double-blind, to placebo or to paroxetine, right, an antidepressant. And the people that got an antidepressant only had a 9% rate of developing major depression. So you give people inflammation, they get depressed. They get the same symptoms depressed people do. You can block it with an antidepressant. It's a pretty nice face value argument that inflammation causes depression. There are hundreds and hundreds and hundreds of studies showing that people with depressive symptoms on average have higher levels of inflammation than people that aren't depressed. Here is one of 2 large meta-analyses that confirm this. You know, basically any lines that are on the right of the arrow suggest that depression is associated with inflammation. And of course, like everything in psychiatry, there's some outliers, but you can see if you were betting on which side had more lines in it, clearly you'd want to bet on this side. It's a very powerful, reliable effect. So there is, in fact, a lot of data that inflammation causes depression, and as you'd predict, depressed people have increased inflammation. That's the least controversial thing I'm going to say. This is much more controversial and more interesting, I think, now. Second proposition is that on the whole, the patterns of increased immune activity associated with depression, so if you make a list of the inflammatory changes that you tend to see in depressed people, these changes should have decreased mortality from infection in ancestral environments. Okay? Now, if you're with me, you should immediately say, wait a minute, there's a problem here. We know depression is not good for your health. Right? If depression was good for your health, we wouldn't be trying to build up this big med psych service and mind, body, brain, cancer. I'm looking at Karen, right? You'd be going around insulting sick people, trying to make them more depressed, so they really kind of rose to the occasion, right? So I don't need to tell you. There's all sorts of data. Let me just take my word for it. Even us. So we did a study where we showed that the more depressed you got on interferon alpha, if you were taking it for hepatitis C virus clearance, the less likely you were to get rid of the virus. Depressed people have changes in their immune system, but they clearly don't do as well in the context of medical illness, and they seem to be vulnerable to infection. All right? So this is a real challenge to this idea. If I'm suggesting that the genes that drive depression, that the genes that drive depression have been selected because they help you survive infection, what gives? Now, I've discovered something in my life, which is if you have a really tough question you're trying to answer, instead of answering the question, see if you can ask another question, right? First off, it confuses people. But second off, sometimes it leads to some interesting ideas. So let me ask this question, that how can this be so if depressive symptoms are so bad for your health, by asking another question, which is this. Do the innate immune inflammatory processes that are increased in depression produce the same patterns of infectious vulnerability and adaptive immune impairment that are apparent in depression? So do you see what I'm saying here? So what I'm asking is not, I'm taking a step back, and you'll see why this is important. And the answer is absolutely yes. There is, and again, there's a ton of literature on this. I'm just touching upon it. There are multiple ways in which chronic inflammation robs Peter to pay Paul. When you run inflammation high, it actually suppresses aspects of your immune system that are essential for fighting infection, including natural killer cells, T cells. So in fact, we know that this kind of chronic inflammatory activation you see in depression does indeed produce exactly the same kind of immune decrements that set you up to have problems. Here's evidence of that. This is an interesting study. They took mice, and some mice got just chronic a thing called LPS, right? It's the cell walls of bacteria. It's a big inflammatory stimulus. So some mice were chronically inflamed, other mice weren't, and then they gave all the mice a shot of the flu virus. And the inflamed mice, rather than doing better, died. That's what you can see here, right? So we know that the types of chronic inflammation you see in depression do negatively impact adaptive T-cell, B-cell functioning, especially T-cell functioning, especially when inflammation is chronic, as it tends to be in depression. And in animal models, you can see that there's a price to pay. It actually does increase vulnerability to infection in some contexts. But the point is, and the reason that I asked that question is, I'm not saying that the genes that cause depression are supermen that save you at all times and all places. All this theory requires is that across evolutionary time, right, so across really many millions of years, because we're not just talking about humans now, we're talking about things that go all the way back into mammals and invertebrates in general, that across evolutionary time, the survival benefits of enhanced inflammatory activity that are characteristic of depression outweighed any costs imposed by associated reductions in other parts of the immune system, right? So you can have all sorts of decrements as long as the overall benefit outweighs them. And there's a guy here named Bruce Ellis who's in the Norton School, who's the big sort of king of mismatch theories. And in fact, part of the problem is that the world's changed so much that we're not benefiting, as I'll show you, from the inflammation. But so the question is it true that running your inflammation high helps you stay alive in environments like the ancestral environments where pathogen loads were high? And there is a remarkable series of studies from Holland suggesting yes. So let me show you some data to support this idea. So Ghana is an interesting country. It's got two different regions that are very different in terms of pathogen exposure. In the Upper East region, infection is prevalent. People still drink polluted water, and most people die from infection. So these guys went in and they looked at various haplotypes, so various combinations of the forms of genes for inflammatory and pro-inflammatory and anti-inflammatory cytokines. And they looked especially at IL-10. IL-10 is a major anti-inflammatory cytokine in the body. And they found, when they looked at different haplotypes in this population, they found that certain combinations of the gene, certain haplotypes, one in particular, was associated with more inflammation. Okay? So people that had this form of the IL-10 gene made more inflammation. And what they found was that the inflammatory form of that gene was more common in young people, in older people than younger people. And of course what that suggests is that it's keeping the old people alive, right? That the people that had the less inflammatory versions of that gene were dying sooner. If you look here up on top, in the modern world, here again in Holland, it's from the same group, you see that with age, generally both anti-inflammatory and pro-inflammatory, TNF-alpha is the classic pro-inflammatory cytokine. Do you see it drops with age? If you look in Ghana, on the other hand, in this Upper East region where infection is just a constant killer, you still see a little bit of a drop in the IL-10, but you see no drop in the TNF-alpha. Now, you can interpret this as that the people are having so many infections that they're still driving their inflammation, or you can interpret it as people that don't make as much TNF-alpha are dying younger. Do you see? But both things point to the same fact. Now, here's the coup de grace on this whole business. So as I said, there's two different regions in Ghana, one where people drink water that's polluted from rivers and wells, and other areas that have been modernized where they have these boreholes where the water is pure. Now, you would predict, if you thought about it, what this idea says is that people that have genes that make more inflammation, A, should be at increased risk for depression, but B, they should survive infection overall if this idea is right. And that's what you see. So over here on this side, what this says is that people that drink polluted water die sooner than people who drink clean water, and you notice that the death occurs all across the lifetime, right? This is going on when you're young also. Remember that infection kills people early in life in sort of traditional societies and probably in the environments in which we evolved. Now, if you look at crossing the pro-inflammatory versus the anti-inflammatory form of that gene, of the IL-10 gene, with what people drink from, what you see is that if you are drinking polluted water and you do not have the pro-inflammatory form of the gene, you die sooner. And the death starts early. It starts in your teens, and you're more likely to die across your entire lifetime. On the other hand, if you've got the pro-inflammatory form of the gene, you're more likely to survive all the way. It's that dark line. Do you see that? Now, here's the amazing thing here, and this explains the entire problem with the modern world. People that were drinking clean water had exactly the opposite finding. If you drink clean water and you have the pro-inflammatory gene, you die sooner, because you don't need all the inflammation and you're paying all the price in terms of tissue damage. So here, and it's kind of hard to see, but in the fainter lines, here's the people that had the pro-inflammatory gene. What you can see, though, is that the pro-inflammatory gene doesn't kill you sooner until you get to be about 70 years old. All right? The reason we all die of stuff in our 60s and 70s and 80s is because the more inflammation we have, the more oxidative stress we have, the more we just eat away at our bodies and brains. But it takes a while. And there's some beautiful studies in the West showing that the same IL-10 gene, that if you have forms of it that make a lot of IL-10 so that you don't have as much inflammation, you're much more likely to live to be 100 in the modern world. So if your inflammation is up in the modern world, you don't really need it because you got hospitals and medicines and you're going to wash your hands and flush the toilet, but you're going to pay the price and you're going to get cancer, diabetes, heart disease, dementia, depression. If you live in a place where the pathogen burden's high, if you live long enough, you still might get all that stuff, but you're much less likely to die at 15, 20, 30, 40 from infection. And so this is very powerful evidence that, to this point, that supports this pathos D idea that people that just generally run their inflammation high, it's like shooting at all comers in high-risk environments, that it's a strategy that outperforms lower inflammatory strategies. Now, I've cut out a bunch of stuff. There's all sorts of studies in the modern world. You can still see the pattern. There's all sorts of evidence to suggest that in many illness states, if you run your inflammation a little higher, you're more likely to survive. So you can still see that signal. But this study is beautiful because you can see the high pathogen, low pathogen, and the difference. And it really supports this idea that if you have a form of it, now this doesn't get to the depression yet, but that the pro-inflammatory, if you have genes that drive inflammation high, you're more likely to survive infection. All right. Well, this is really the main point, right, which is that allelic variants, so the forms of genes that increase the risk for depression, should enhance host defense mechanisms in general and innate immune inflammatory responses in particular. So what I'm saying is something very simple. You give me a gene that's reliably associated with depression, and there are none, which is a problem with this whole idea, right? But if there were, you give me the most reliably associated genes, and I'll show you a gene that, at least in ancestral environments, contributed to protecting you from infection, all right? Or at least some fair number of depressogenic genes should have that characteristic. That's the clear idea. So to be fully consistent with this pathos D idea, the risk forms of genes for depression should meet at least the following criteria, right? So they should be located in genes with known immune effects. This does not mean genes of the immune system. It means that if a gene is associated with depression, that gene is going to have an effect on the immune system. And all you have to do is go looking for it. All right? And in fact, I'm sparing you I got a table that's 15 pages long with 400 references where I basically pooled all the top genes that have been associated with depression, and indeed about 95% of them have clear immune effects, and many of them have clear effects on infection. 2, genes associated with depression should increase signaling in inflammatory and host defense pathways in general, right? 4, genes that increase your risk for depression should increase survival in the context of infection. And 4, whatever genes you look at should be supported by the best evidence available. And so in that light, let me show you some of the best known data. So, you know, there's different ways of looking at whether genes are associated with an illness. The most rigorous way is a thing called genome-wide association studies, right, where they look at hundreds and hundreds of thousands now of different variations across the whole genome and just say, are there any differences between this group of depressed people and this group of not depressed people? Now, it's like flipping a coin 1,000 times, right? If I flip a coin once and I say, you know, what are the chances that it's going to come up, that it's not going to be tails? You say 50%. What if I flip a coin 1,000 times and say, what's the odds that it didn't come up tails once? Right? Well, it's almost nothing. In the same way, because of that, if you're going to look at 100,000 things at once, you have to do so much correction for that flipping the coin multiple times, that only genes that are really, really associated with something are easy to find. And so they're not so easy to find depression genes. But if we take candidate genes, this is a looser approach, where you think, you know, serotonin's got something to do with depression, so here's a gene, let's see if it's associated with depression. It's like a candidate, right? Those studies they're easier to show effects, but they're not as reliable. So if you take candidate gene studies and you say, do any of these survive in this large GWAS type fashion? So this is the toughest way that you can test a gene that you think is associated with depression. In fact, there's only one gene and really 2 other SNPs that make the cut. What this tells you is that we know almost nothing about the genes associated with depression. I gave this talk to cardiology, and there's a world-famous genetic researcher came up. She said, man, Chuck, that's really interesting. The only problem is we don't know anything about the genes that set you up for depression. And this woman, that's her whole life's work. So this really, first off, it points that we know almost everything about environmental factors that cause depression. And it never ceases to amaze me that our jaws should drop at the gap between how much we understand about the environment and how little we understand about genes, and how much we go on and on about genes. But that aside, these are the 3 most likely SNPs from candidate gene studies to be associated with depression. And what's interesting is that all 3 of these have clear effects and roles in the immune system. Now, I have about 8 slides that go over the effects of all these. I'm going to skip that. And let me give you another, because I want to show you 2 other slides, just an example. Here is another way of looking at whether candidate genes are associated with depression. It's not as good as looking at a GWAS. This is a meta-analysis, right? So they took all the studies that have ever been done that are worth looking at and saying, do any of these candidate genes survive in a meta-analysis? And some do, and I want to just point out 2 to you. First, this GNB3, and here's the short form of the serotonin transporter, okay? What do we know about these genes and the immune system? So let's start with that GNB3, and I'm only going to show you these 2 genes. So it turns out that the form that's associated with depression, it's 825T, produces a variant of this gene that is a gain of function variant. This gene is involved in G-coupled signaling, so it's in signal transduction in cells. The version of the gene that is associated with depression increases the activity of this gene, right? Now, if this gene is increased in depression, and if depression is an inflammatory condition, this gene should have pro-inflammatory functions when increased, and indeed it turns out that the form of the gene that's associated with depression increases immune activity, increases inflammatory activity, just as you predict. Now, this is really interesting. And again, I could give you something similar for probably 60 to 80 genes. I'm just picking a couple, right? All sorts of evidence for this one know that it has these very powerful immune effects. People that have the depression variant also have evidence of hyperinflammation, right? So there's other stigmata that people with this, that carry this form of the gene, for instance, they have cardiovascular disease, hypertension. And remarkably, the form of the gene that's been associated with depression has been shown in many studies. It reduces death from infection in infancy, and that's the primary thing, right? So right there, this is a gene that meets all the criteria that would suggest this idea that a gene that's known to drive depression, or at least we have a pretty good idea it drives depression, activates inflammation, and there's a signal that it keeps babies from dying in infancy. The more pathogen exposure there is around the world, the higher the prevalence of this gene in those areas, right? And then there's all sorts of interesting data that the allele that's associated with depression increases it gives you a better antiviral response to interferon alpha. It improves responses to retroviral therapy and HIV. And if you've got the depression variant, you get a better antibody response to hepatitis B virus. So here's a gene that's very interesting that is a depression risk factor, and it meets all these criteria to suggest that that form of the gene also enhances host defense, right? Or at least enhances host defense in some conditions, as we'll see. Now, I want to talk for a second about the serotonin gene because we all know about it. If there's any gene that's associated with depression, this is probably it, even though it doesn't come up in the big GWAS studies. But there's a very good data set that if you have the short allele, you have a hypersensitivity to environmental conditions. If environmental conditions are good, you prosper more. If environmental conditions are bad, you suffer more. Right? This is the idea of the orchid child. This is the classic orchid child gene. The neuroimaging on your left shows that you can take non-depressed 8-year-olds and little 8-year-old kids that have the short form. If you show them sad faces, their brain activates more, and it activates in the areas that are increased in activity and depression. And then the graph on the right just shows that this is a meta-analysis of all studies that have looked at it. There's a pretty powerful association between the short allele and getting depressed if you're under psychosocial stress. Okay? This gene if it induces depression, it should have immune functions, and it should be a pro-inflammatory gene, is it? Well, there's two studies that say that it is. This is one of them. This is an interesting study from Stanford, from Fergus DeBar. He took women that were not depressed, they were medically healthy, put them through a stress test in the laboratory that we use all the time called the TSST, and showed that if you looked at the ratio of IL-6, which is pro-inflammatory, to IL-10, which is anti-inflammatory, women that are not depressed, that are otherwise normal, if they have the short allele, they run their inflammation high. They run their inflammation high at baseline, and they run their inflammation high in response to stress. The woman who came up to me after the Emory lecture and said, great idea, but we don't know anything about the genes that cause depression, showed the same thing, that if you have the short allele, you run your IL-6 high. Okay. More amazingly, the short allele, this nasty allele that we think maybe we should genetically engineer, turns out to protect you from sudden infant death syndrome. There are several studies showing that the depressogenic variant decreases the chance of infants dying of SIDS. Now, SIDS is not always an infectious condition, but many, many times it is. And even when you can't find the organism, almost always there's this strikingly increased inflammatory signaling going on in SIDS, suggesting that there's some kind of occult infection that we're missing. So here's an example of a gene that, not good, causes depression, we think, but it's also pro-inflammatory. It also seems to have effects on survival in infectious relevant disease states. And then my all-time favorite, remember I said that the short allele of this serotonin transporter gene, 90% or so of the people in Japan have it. Well, it turns out that people have gone through, independently of any of this, there's a whole registry on how much pathogen exposure societies have had over the last 1,000 years or so. They call historical pathogen exposure. If you cross historical pathogen exposure by the prevalence of the short allele, what you find is that the more bugs people have had to cope with, the higher the presence of the short allele. Now, I can't go into this today, but we think, everybody here thinks the short allele produces depression. On a national level, the nations that have the highest prevalence of the short allele have the lowest rates of depression. And that's got to do with the immune system and host defense also. It fits into this story, but I just can't go there. But here is evidence then of this risk gene that you can even see it on an international level. Okay, and then finally, depressive symptoms should overlap with the symptoms of sickness, right? So this is really an important point. I am not, in fact, saying depression is like sickle cell disease. What I'm saying, in fact, is that the genes that produce depression, genes that produce inflammation produce depression. Inflammation produces depression. Depression is really, let me just skip ahead for a second, is really just, in some ways, another word for sickness in response to stress. If you look at the symptoms of sickness and depression, they overlap so strikingly that what I'm suggesting is these genes produce a behavioral and physiologic state, and that state is absolutely essential to the way that those genes fight infection. Sickle cell disease is a little different, right? It's just kind of accidental that the blood cells get kind of funky, and so the malaria can't handle it. This is really a more striking thought, right? What we're really saying here is that the genes that cause depression enhance pathogen defense. That's why they've been selected and maintained. One of the ways that they enhance pathogen defense is by producing depression, because the symptoms that we call depression have anti-pathogen abilities, all right? And there's a good 3 hours to go over all the symptoms of depression and show how they might help keep us alive in the context of depression, of infection. When we think of sickness, in the old days, people used to think about it a lot the way we think about depression now. It was seen to be a bad thing. Does anybody here like throwing up, having diarrhea, having shakes, feeling achy? No, of course not. But we know now that those symptoms evolve because they have anti-pathogen properties. The symptoms of sickness exist to help the host defeat the invader. The classic examples of that are fever and anemia, right? So fever is sort of the classic central symptom of bad infection, and fever has been shown just repeatedly, as you can see by all these references, to help battle both viral and bacterial pathogens, right? And it does this over and above the effects of the inflammatory pathways themselves. So inflammation does stuff to kill bugs. On top of that, fever kills bugs. And there's data in animals that if you block fever, they're more likely to die from infection. Nice human data showing you get the flu. If you start taking nonsteroidals, you will not be as sick, but you'll be sick longer. So even in humans, you see the trace of the fact that these symptoms, especially fever, has pathogen-fighting abilities. So not only does fever cause microorganisms to sort of denature and fall apart, but it also ramps up the ability of the immune system to function. The immune system actually works better when it's a little bit heated up. Now, it's also interesting that the effects of fever are even more powerful in conditions of low iron availability. So it shouldn't be surprising that in addition to causing fever, inflammatory cytokines, inflammation, this thing that's up in depression, deplete bodily iron stores. If you're inflamed, you're not going to absorb iron, and you're going to get rid of the iron you have, and you're going to sequester it. And Ashley's nodding. She's interested in this stuff. The reason it does is because bugs need the iron as much as you do, right? Especially bacteria. It's a way of trying to starve out the bugs, right? So very distressing data from around the third world suggesting that it's probably good to give little kids iron, but if they're in an environment with a lot of pathogens, the price they're going to pay is more infection. So here's a study from Kenya showing that this is the percentage of kids that get infected. If they have fully repleted iron, it is almost 3 times more than if they're mildly iron depleted. All right? And it's been shown in a number of studies around the world. You can give the kids the iron, but you're also feeding the bugs. Right? So very clear evidence that reduced iron and fever have these anti-pathogen effects. Well, I didn't go into about stress and wounding, but because we know stress is a major risk factor for depression, there's a lot of evidence that our stress responses try to help us cope with wounding to avoid the infection that comes with that. Stress should do the same thing. And there's plenty of data that psychological stress causes fever in human beings. This is data from Japan looking at psychogenic fever. It causes it more often than women than men. Of course, women are also more likely to get depressed. And it causes it in the young ages, prime reproductive years, when depression is also at its prime. This is an amazing study. This is back before they had high-sensitivity CRPs. It's done in 1991. They took a bunch of military recruits before and after Hell Week, which is this 5-day thing where they don't sleep, they don't eat, they're tortured, you know, they run them through psychological hell. After Hell Week, even days after Hell Week, huge increase in C-reactive protein, but for today's point, a lot of things down, but iron is down almost 50%. So stress reduces iron. We know from animal studies that stress reduces iron. Major depression is a febrile illness. It's been shown in repeated studies. My favorite by Marty Zuba. He actually took depressed people up here and got them to wear a rectal probe for several days, which is remarkable. I started my research career with rectal probes, and I had to abandon it. I still do LPs because people are okay with that, but the rectal probes, I couldn't get anybody to wear them. We tell them we wore them, it's not a problem, just didn't work. But anyway, and I knew guys in the study that were in the control group. People with major depression run several degrees Fahrenheit hotter across the entire circadian pattern. The amazing thing about this study was Marty Zuba, who's passed away, God rest his soul, then gave all the depressed people ECT and measured their core body temperature again, and swear to God, that's what happened. Once the depression remitted, the body temperature was indistinguishable from the normals. This is only one of a number of studies. There are also several studies, although not as many data sets, showing that depression is also a state of reduced iron availability. Now, here's the thing, because fever and reduced iron are central to the adaptive purposes of sickness, their presence in depression is really mandated from a pathos D perspective, right? If depression did not share those sickness symptoms, you could pretty much forget this idea. On the other hand, and their absence argues against validity of this approach. On the other hand, it's not so clear. If depression really has to do with your psychosocial interactions, not so clear why you'd see reduced iron and febrile body temperatures in depression, right? It just doesn't make any sense at all. So this is very I think this points powerfully to the fact that the primary reasons of depression may have more to do with our relationships with pathogens. I'm going to skip this part. Let me just make this final part, and then I'll quit. Too bad. It's really interesting. This is all about little turkey poults. It turns out that if you're a really turkey baby that's on the move, and it can kill you from infections, but that's a story for another day. Let me end with this. This is really what I'm trying to say. Major depression is a relationship disorder. We all know that. I don't need to tell you that. We know there's genetic vulnerabilities. We know some people do things that ruin their relationships. It's got a profound interdependent nature to it, right? All I'm saying is that if you want to look at its adaptive purposes, we need to expand the realm of interdependency, that it does not have to be just adaptive vis-a-vis other people, that in fact the data are better, that its adaptive purposes is in our interrelationship with the microbial world and the parasitic world. Now, as with humans, in which everything is a give and take and a dialogue, so with immunity. One of the great fallacies of the psychoneuroimmunology field is it looks at inflammation as if it's something we just produce. We're eating the wrong things, we're not getting enough sleep. But that's not really the essence. It's really about a dialogue with pathogens. And I want to give you an example, and I'll close with this. So here is a gene. This, believe it or not, is probably the best documented gene that's associated with depression. How many people here know about this gene? Ever seen it? Isn't that amazing? Yeah, I hadn't either. CACNA1C codes for a type of L-gated calcium channel, the alpha-1 subunit. In these big GWAS studies, this is the thing that most often comes up. It doesn't come up most often because nothing comes up most often, but this is probably the best runner-up for a depression gene. The people that have the A allele of this RS10, this one, the people that have the A allele, the risk allele, at this point in this gene, also, if you just look at normal folks, their brains are changed in ways that you see in depression. The hippocampal functioning is not as good. The connection between the amygdala and the hippocampus is suboptimal, okay? So there's convergent data to suggest that this gene does something that's related to depression. Calcium signaling, which is mediated by this L-type voltage-gated calcium channel, is also integral to all sorts of aspects of immune system. This channel that this gene codes for contributes to the function of a whole bunch of different immune cells. So here we go again. Here's a gene that we know involved in brain function. It also turns out to be very involved in the functioning of the immune system. And if you look at this channel, if you block it with calcium channels, inflammation goes down. So this suggests that maybe this particular calcium channel is pro-inflammatory. If that's true, then you would predict that the risk allele for depression should be a gain of function variant. If this is a pro-inflammatory channel and major depression is a pro-inflammatory condition, the gene, the form of the gene that sets you up for depression should make more of this channel. There's some evidence for that, actually. And there's also evidence that this channel, that people that have this depression risk allele, we know they make less of a very important messenger that is involved in suppressing inflammation. So there's some pretty good data to suggest that the form of the gene that's been associated with depression has pro-inflammatory properties. And we know that this channel, which is hyperactivated probably in depressed people, is very necessary for T-cell defense against a bad infection called Leishmania major. Okay? So that makes a nice story. Give people calcium channel blockers, their inflammation goes down. There's some evidence that people that have the risk factor have more activity through these calcium channels. That activity probably helps you with at least one bad bug, but there's a fly in the ointment. There's a disease. It's not the same gene. It's not the same place in the gene, but there's another disease which is clearly caused by an increase in the activity of this calcium channel. It's called Timothy syndrome. These poor little kids have a bunch of problems, but one of the problems that's relevant for now is that these little guys have huge increased risk of infection. So here's a disease state, increase in this channel, increased infection, not decreased. And it turns out that this channel actually fouls up your ability to fight tuberculosis. And so there's this conflicting evidence to suggest that if you activate this channel too much, it might actually put you at increased risk for these very serious infections that have been with us, since time immemorial. And this is what this gets at. Both are probably true. When we think about immunity, just as we think about our interactions with other people being like a conversation, the immune system's like a conversation too. It has to do with how we interact with bugs. And bugs are not passive. They've figured out ways to manipulate our host defense. So there could be genes that code for inflammation that in general keep you alive in the face of certain infections, but other sneaky little infections will figure out ways to subvert those very defenses and use them for their own advantages. Here's some classic examples, right, of pathogen manipulation. Here's that ant. Remember I said the ant that had the little thing coming out of its head? That fungus takes over the ant's brain and marches the ant up to the tip of the grass, where it grabs on and waits. And the reason it does that is because the sheep come along and eat the grass. And the bug needs the guts of a sheep to reproduce, even though it's the death of the ant. Rabies, of course, we know that is a classic example. It makes mammals aggressive and drool so the virus can spread. But here's my favorite example. They actually, great study. People got the influenza vaccine, and it turns out that if you get a flu shot, it makes you social. So what they found was people were much more likely to be out and about, going to a party, whoopee-whoopee, in the couple of days after getting a flu vaccine, at exactly the time, whereas if it was the actual virus, they'd be most infectious. Now, once the infection hits, forget it. You know, anybody here ever had sex when you were thrown up and crawled to the bathroom? You know, probably not. But when you're really, if you have, I asked that question. I'm trying to collect stories for this paper. Come talk to me afterwards. It'll be my private psychiatric confidentiality. But before you go down, this is a classic example of pathogen manipulation. The influenza virus manipulates our behavior to make us more pro-social so it can spread. So when we think about immune system, we need to think conversation, interaction, interdependent relationships. Back to the CACNA1C. Well, okay, so it turns out that that this channel is absolutely necessary for immune cells to kill certain organisms, like this chlamydia pneumoniae, right? If this channel is not working, you're going to die from the infection. But on the other hand, other microbes, in this case a Legionella, have actually evolved to take advantage of this same channel, and if that channel doesn't work, they can't reproduce, right? So here's the point. This is what it really says. So if the risk allele at this particular, the calcium channel, if the risk allele continues to be identified as a risk for major depression, Pathos D theory predicts that its overall effect on immune function should be stimulatory. So on the other hand, if it turns out, following this logic, if the A allele is confirmed as a gain of function variant, then Pathos D theory predicts that the summed effect of the L-type calcium channel will be immunostimulatory, precisely because it is associated with depression. Here's the point. You can work this idea backwards. If you look at the genes that cause depression and the illnesses that they protect against, that is a way of figuring out how devastating any given illness has been in a given human population, because the high price of depression has to be balanced by that effect, right? So what you're really going to see is that depression genes help you in some illnesses and maybe hurt you in others, because pathogens figure out ways to get around these mechanisms. But the genes that cause depression that are common, whatever they do to the immune system is a trail of strategies that overall have kept us alive. So you can actually use this theory to work backwards to begin to understand something about our interactions with microbes over time also. Okay, well, I'm sorry I've gone too long. Let me stop. But I hope that gives you a flavor of how something that we don't tend to think of as being part of our psychiatric world, in this case immunology, can really shed new light on conditions that we all have to deal with every day. So, okay, thanks. Any questions? I know people